So, I'm V, and I study epidemiology with a focus on infectious disease. Today, I'm going to be doing a talk on coronavirus epidemics and their investigation using molecular epidemiology. I'll focus on more like the origin and what molecular epidemiology can tell us about the transmission of those viruses, as well as how the epidemic started. More on that aspect than, on specific numbers of cases. I won't touch on numbers of cases per month or anything like that. So first, before I start, I want to give thanks to my professor, Konstantinos Chamis. He was my instructor for a class, and this presentation is based on the paper I wrote for that class. So everything is taken from that, all the text and the figures, all the work for this that went into this presentation is taken from that paper. And I will also want to thank Roger Frutos. He is a French infectious disease researcher, and I cited some of his work for this paper in the presentation. So I also talked with him privately, and he helped me. He gave me some valuable insight on the topic of coronaviruses. I also want to thank Enigma, because she was very patient with all my time going into this work. Right, as I said before, I talked about the purpose of this paper, but to be more clear, I will describe the search for the origin of the amazing coronaviruses of the subgenus Sarbecovirus. And I will explore hypotheses and theories regarding their origin, the origin of the epidemics, that is, the compatibility of those theories with the available scientific data. I will describe the course of research from the beginning of the outbreaks to the present day, like all the information we have from the beginning of each outbreak to now. I will not be touching on the MERS epidemic, as the MERS coronavirus belongs to the subgenus Merbecovirus and not Sarbecovirus. So these are the contents of the presentation. First, we'll talk about some basic terms that are related to this topic. Then we'll talk about the first Sarbecovirus outbreak, the SARS outbreak of 2002. People who are older than about 20 years old will probably remember that, as it made the news, the global news. A lot of people haven't heard of it, even those who are older than 25 years old. Then we talk about the molecular epidemiology and phylogenetics aspects of that outbreak and investigating the origin of the etiological agent that caused it through those fields. Then we talked about the ongoing COVID-19 pandemic. I said outbreak because it began as an outbreak and then it evolved as a pandemic, just like every pandemic does. It's not a pandemic initially, it begins as an epidemic or as an outbreak, and then it evolves to a pandemic. We'll talk about some theories that have been proposed about the origin of the etiological agent of the COVID-19 outbreak. Two of those theories with the current scientific data, and then we're gonna much have a discussion about conclusions that we can derive the available data. I mistakenly put acknowledgement here at the end. It was initially at the end, but I moved it to the beginning because I thought it was more fitting. Right, let's begin. Introduction. According to the WHO, zoonotic diseases, also known as zoonoses, are defined as infectious diseases that are transmitted from animals to humans. Zoonotic pathogens can infect humans after contact with an infected animal. Such events can give rise to epidemics which may develop into pandemics, as is the case for the outbreaks that we'll talk about here. The pathogens that cause zoonosis are diverse and can belong to different taxonomic categories. That means they can be bacteria, they can be viruses, they can be protists, and they can even be animals. A lot of parasitic worms are actually animals, are classified in the animal kingdom. So when you think of a pathogen, it can be pretty much anything from a virus to a bacterium to a parasite. I give an example here. The West Nile virus is a virus transmitted by certain mosquito species, and it is classified under the family Flaviviridae, and it causes a disease called West Nile fever. Now, you'll see me, I give emphasis on the distinction between disease and etiological agent or pathogen. The disease is called West Nile fever, and the pathogen that causes it is called West Nile virus. I think that distinction is important, although sometimes the disease is named after the pathogen that causes it. So they may have similar or even the same names, but a lot of the times they are etymologically distinct, and I think that's quite important, especially in an academic context. So this is a figure that was made with data from the World Health Organization and the International Organization for Animal Health. This is just to put into perspective how important zoonoses are. You can see here 60% of existing human infectious diseases are zoonotic, meaning that they come from animals, they have a zoonotic origin. And at least 75% of emerging infectious diseases, including Ebola, HIV, Ebola virus disease is the disease. The disease caused by HIV is known as AIDS, and influenza have an animal origin. And a lot of the diseases you might have heard of have an animal origin. 5 new human diseases appear every year, and 3 are of animal origin. 80% of agents with potential bioterroristic use are zoonotic pathogens. And I got this figure from Dr. Bourré. He's the director of the Department of Global Health at the Institut Pasteur. So thanks to the credit goes to him. I didn't make this. It's a very important one. Health is a very important approach that isn't been talked about in pop culture or a lot, even academically. I've interacted with a lot of people who study relevant fields such as microbiology, and they are unaware of what One Health is. I think it should be taught in schools, and generally more people should be aware of what it is, because as you'll see here, it's quite important. So let's begin by defining public health, which is the science and art of preventing disease, increasing life expectancy, and promoting health through organized community action. Well, One Health refers to the integrated approach of human and animal health, including food safety and the environment, because as you'll see further into the presentation, all those aspects of health, human, animal, and environmental health, are integrated, and you'll see why in a minute. One Health is a multisectoral approach that allows for the fields of human medicine, veterinary medicine, environmental sciences, public health, and epidemiology to collaborate in order to address global issues. So you'll see it's a very multidisciplinary field of study where many professionals from the relevant fields cooperate. It's mainly veterinary medicine. A veterinarian coined the term One Health, so that's how it began, but it integrates both animal health and human health, right? So this is what's known as the One Health Triad, the classic circle where you see it's like a Venn diagram almost. You see human health and animal health and environmental health, and they all meet in the middle, which is what One Health is, because it's we refer to as the human and animal interface, because human and animals interact with each other, and knowing the definition of zoonosis, which are diseases and transmitted from animals to humans. This is why One Health is important, because humans and animals interact and can be interplay between the diseases there, right? It can be cross species transmission, stuff like that. So you see, it's about risk management and public health, disease prevention, disease surveillance, and all that relevant stuff. These are some of the issues that are studied by One Health professionals. It's not talked about as much as it should be, but people who study microbiology would know that, and public health would know that antimicrobial resistance is a very important issue, and it's being ignored, unfortunately, but it's rising. It's a rising issue, and WHO predicts that it will be one of the top causes of death by 2050. It will be probably like the main cause of death. Environmental damage and how it affects human health, such as pollution. Air pollution can lead to detrimental health outcomes for humans and animals alike. Like you can get a lot of lung diseases, such as chronic obstructive lung disease. It's a disease that's affiliated with pollution. Animal diseases, human diseases, and zoonosis across the human-animal interface that I talked about before, and you'll see more about in a few minutes. Vector-borne diseases such as malaria is a classic vector-borne disease, and the vector that carries it are mosquitoes. And now we're going to talk about some basic coronavirus virology, so you know the fundamentals of coronaviruses and how they work, or what they are made up of, because it's useful in order to understand the presentation. So the term coronavirus refers to Coronaviridae, which is a family of positive sense single-stranded RNA viruses. Now, if you're not familiar with molecular biology and the Baltimore classification, just keep in mind that they are RNA viruses. I just added in a few extra details for the nerds here, alright? But if some of that stuff confuses you, just keep the essence. They're RNA viruses, and they belong to the class 4 of the, according to the Baltimore classification system, which is a system we use to classify viruses based on their molecular biology and how their genetic material is copied and how they reproduce. So coronavirus particle, which is called the virion, it's virus particle, no matter if it's a coronavirus or Ebola virus, it's called the virion. A single virus particle is called a virion. That is an explanation for, any people have question as to what virion is. So it's spherical in shape, like a classic virus particle you see portrayed often, and consists of a nucleocapsid, which is a complex of a nucleocapsid protein along with the viral genomic RNA, the genomic material of the virus, viral envelope, which encloses the above complex, and other structural proteins are part of the viral envelope. The spike protein, membrane protein, envelope protein, and there are some other proteins in different coronaviruses, but those are the basic ones that concern us here. So the spike protein, you might have heard of it because it's quite important. It mediates cell entry, and it's usually composed of multiple subunits, like there's the S1 subunit and the S2 subunit. and it's a trimer. In SARS coronaviruses, it's a trimer. That means it consists of 3 subunits. And as I said, it's the main protein involved in viral cell entry into the host cell. Coronaviruses are a family of viruses of health interest since they've been the cause of several epidemics, as you'll see in a few minutes more detailed. So this is a picture I made using BioRender. It's basically to give you an idea of what they look like. You see all the parts I mentioned above. You see the nuclear capsid in the middle, and the viral envelope is the circle thingy that you see, and the spike protein are the point-like structures, and that's what the virus uses to infect the cells. It binds to a receptor in the cell, in the host cell, and enters via receptor-mediated cell entry. So we're going to talk about the first and the original coronavirus outbreak, which was in 2002, the SARS outbreak. So in late 2002 and near the beginning of 2003, cases of pneumonia of unknown etiology, otherwise known as atypical pneumonia, started being detected in Heian City, Guangdong province in China. Following an epidemiological investigation by the Guangdong CDC that began after the initial cases were reported, it was discovered that there were cases in 6 other municipalities in China from November 2002 mid-January 2003. So cases started being detected in early January 2003, and after a lot of cases were discovered, the Guangdong Center for Disease Control found out that there were cases since November, so it had been going on undetected for a few months. Right, so the World Health Organization was notified in mid-February, about a month after the initial cases started being detected, and the disease was named Severe Acute Respiratory Syndrome, otherwise known as SARS. On March 12th, the World Health Organization declared a global alert on cases of atypical pneumonia of public health concern in China, Hong Kong, and Vietnam. It had spread to 2 other countries in March 2003. Now, epidemiological and clinical data from the beginning of the outbreak suggested that this disease was infectious in nature, and the most likely etiological agent was a virus. They didn't know what was causing the disease, but data suggested it was an infectious disease and it was caused by a virus. Rigorous epidemiological studies and laboratory investigations of the samples from the patients led to the discovery of a new virus, which was identified as samples from patients diagnosed with SARS, but it wasn't isolated from control groups, from control patients in studies that had a control group. And those patients who had SARS, who the virus was isolated from, tested negative for common pathogens associated with common respiratory tract infections, such as Mycoplasma pneumoniae and all the other common pathogens that we test when we suspect a respiratory infection. That is important because it rules out that there are other pathogens causing the infections, and it gives you an idea that perhaps the novel virus you isolated is responsible for the outbreak, because they didn't know it was a coronavirus yet. The data from molecular analysis of the virus, including some techniques I've mentioned here for the NADs, which is, for example, reverse transcriptase PCR, ELISA sequencing, and BLAST and phylogenetic analysis, so that the new virus that was isolated from patients with SARS begins with the family Chronoviridae, which means it's a coronavirus, and it was named SARS coronavirus after the disease. Epidemiological evidence suggested that the virus is transmitted by direct contact through respiratory droplets like sneezing and through contact of the virus with the mucous membrane. So sneezing or wiping your mouth with hands that are infected with the virus, that have come in contact with fluids from an infected patient. That's a route of transmission. So here we're going to talk about investigating the origins of the first coronavirus epidemic, SARS, using molecular epidemiology and phylogenetics. So to begin, a large proportion of the early patients were food handlers, and that means they were involved with handling, slaughtering, and selling food animals, or preparing and serving food. That includes butchers, it includes chefs, and other workers that handle food in their occupation. And they worked in places where animals intended for human consumption were slaughtered or sold. A lot of the chefs handling food animals in China, in those places, were bringing live animals to the restaurant, and they slaughtered the animals on the spot. So that's important in order, that tells us some clues about the disease. The index cases, which means the initial cases for 2 of the first 7 cities with SARS cases, were chefs who handled exotic animals. It is important to note that in China, at least in those regions of China, some exotic animals were considered a luxury food item. One of the index cases whose symptom onset was in early January was a snake vendor who sold exotic animals, among other things, at a wet market. And he died about 21 days after the symptom onset. The first identified SARS patient who was identified retrospectively, that means after the outbreak started. He wasn't the first diagnosed patient, but he was found after the outbreak started. He had symptom onset on mid-November, and he prepared food from animals, which might give us some important clues as to how the outbreak came to be. And he had no travel history, which means he couldn't have gotten it outside of Foshan municipality, which is where he lived. So that tells us that the disease originated close to that region and it didn't come from outside of China. And he was the source of a cluster of 5 patients, which doesn't tell us much about the origin, but it's important to note, so I mentioned it here. The later clusters in Foshan municipality started from food handlers, which tells us gives us some clues about the origin. Maybe food handlers are the source of this virus, or they are associated with it somehow. Guan et al. So for non-academics, et al. Means and others. So when I say Guan et al., it's a study, it refers to a study led by Guan, that's the first author, and other scientists who worked on it. In 2003 is the year that the study was published. So Guan et al. In a study in 2003 isolated and described coronaviruses related to SARS coronavirus from animals, mainly the Himalayan palm civets and raccoon dogs found in a live animal market. And they used techniques such as RT-PCR and antibody tests, serological tests, and with Western blotting, which is a technique using antibodies, neutralizing assay, which is another technique using antibodies. That showed evidence of SARS-CoV-2 infection in the animals. Two coronaviruses, SZ3 and SZ16, were isolated from civets, and they were fully sequenced, and they had about 98.8% homology with the human virus responsible for the pandemic, SARS-CoV. That's so that human and animal SARS-related coronaviruses are genetically related. So, but it's important to know that animal viruses retained a 29 nucleotide sequence that wasn't found in most humans, most human viruses. So this is a phylogenetic analysis from Guan et al. It's just for people who are interested in more detail. This is where the animal viruses are placed. I don't know if you can see here. If you can see SZ3, SZ16, and these are the human SARS-CoV viruses, all the different strains. This is the Toronto 2 virus, and this is the Urbani strain. But all these are SARS-CoV-2 virus. I believe this is the original one from Guangdong. It was named GZ1, and those are viruses variants that branched out from that. And these are the animal ones isolated from civet cats. So you can see they're related. That gives us some clues about the origin of the virus. Maybe it's a zoonotic virus, but that's beyond definitive. It's not, well, it's not definitive. So the investigation just started. It's not conclusive yet. So Yu et al. In a paper in 2003 conducted a serological study, that means with antibody testing, and they found that 13% of animal handlers who hadn't been diagnosed with SARS were seropositive, compared with 1 to 3% of individuals belonging to 3 control groups. They had 3 control groups, and they saw that animal handlers, there was a higher prevalence of SARS in animal handlers compared to other unrelated groups. And I'll specify what the groups are later on. And Guan et al., the guys from the previous study, found that 40% of wildlife handlers had, and 20% of animal cultures were seropositive for SARS-like coronaviruses, while none of the patients in the control group came out seropositive. That shows us that there's a high prevalence of SARS coronavirus or SARS-like coronaviruses in people who interact with animals. And that gives us a clue supporting the zoonotic origin. So you can see here with more detail the tables taken from those papers, where you can see here is from the first paper, Guan et al. 2003. You can see that the 3 control groups, here are one control group is a vegetable trader, and there are patients that were hospitalized for diseases other than respiratory diseases, which means that there wasn't any bias detected for SARS or other respiratory pathogens. So random patients in the hospital are compared with animal traders and other people who interacted with animals, and it was found that there's a higher prevalence in people who interact with animals compared to random patients or vegetable traders. That gives us clues again to the zoonotic origin of the disease. And you can see here from the second study, you can see the antibodies. It was an antibody study, just like the first one. Both are seroprevalence studies, otherwise known as antibody studies, of IgG antibodies for SARS-associated coronaviruses. So it's a bit more general here. They have 3 control groups. They have Guangdong Center for Disease Control workers. They have healthy adults at a clinic, and even hospital workers who were more likely to have SARS coronavirus. And you see that animal traders, there's a high prevalence here, a higher prevalence in animal traders than all the other groups. So that is quite suspicious, right? Why is a higher prevalence in animal traders? Gives us some clues about the origin of the virus. And then in 2005, about a year after the outbreak ended, it ended in about 2004. So in 2005, in a publication by Lee and co-workers, it was found that bats of the genus Rhinolophus, otherwise known as horseshoe bats, have a high prevalence of SARS-related coronavirus infection, and concluded that bats are natural hosts and reservoirs of SARS-related coronaviruses. Now I've underlined Rhinolophus, or horseshoe bats, the genus horseshoe bats, because you have to remember that. You have to remember horseshoe bats. They play quite an important role in this presentation, and you'll see them come up a few times. So they found that bats were the natural hosts of SARS-related coronaviruses. This is just a bit of an umbrella term, but you can also use it as a species, and it contains pretty much viruses, coronaviruses, that are related to the original human SARS coronavirus. So you can think of it as a species that contains the original human SARS coronavirus, as well as other non-human SARS coronaviruses that are genetically related to it. So they found, in more detail, they found that 8% of the specimens in the bats they tested in the species Rhinolophus personi from the Guangxi region of China. About 33% of the samples in bats of the species are pusillus from the same region, Guangxi in China, and 71% of the samples in bats of the species are macrotis from the Hubei region in China, positive for antibodies against SARS coronavirus, which it doesn't necessarily mean that they hosted SARS coronavirus, but rather it could mean that they hosted a genetically or an antigenically related virus that they had immunity to, they had antibodies against, because there was prevalence of similar viruses in bats in that populations. So you see serological findings were verified. Serological refers pretty much to the antibody studies. So these were verified by PCR, which is direct detection of the genomic material of the virus. 5 fecal samples tested positive. That means they found the genetic material of the virus. That's one of the most direct detection techniques, because antibodies don't tell you if someone is currently infected with the virus. They tell you that someone has been exposed to it. PCR tells you whether someone has the virus still currently. So they found that they found 5 fecal samples from bats tested positive, 3 belonging to the species R. personi, 1 belonging to the species R. macrotis, and 1 belonging to the species R. fermquinum. So after analysis and sequencing of the PCR products of 1 sample from a bat, it was named SARS-like coronavirus RP3. It was found that it have about 94% homology with the human SARS coronavirus. This strain is thought to, but it doesn't really matter. It's just it doesn't matter what strain it is, because I think the original strain, the GZ01, had even more homology with that bat coronavirus. So I don't really know why they chose this strain from Toronto, Canada, because the original strain, the original human strain from Guangdong, China, had higher homology. but I'm just reporting the findings here. They had 94% homology, excluding the 3 variable regions, because of course those are bound to chains through selection, pressure, and evolution. And phylogenetic analysis with SARS-related coronaviruses and the new samples showed that the human SARS coronavirus is more closely related to those of bats. So that's quite interesting, that a human pathogen is more closely related to bat viruses than other human viruses. This is why the bats are called horseshoe bats. I wanted to include a picture here to explain where the name came from. If you can see my cursor, you can look at the nose of the bat. You see this shape, this rounded thing, kind of looks like a horseshoe. That's why they call them horseshoe bats. It's characteristic of the genus Rhinolophus, and all the species in the genus look kind of like that. And I think this horseshoe part of their nose, it plays a role in echolocation, but I think its role isn't really understood, but that's quite relevant here. So the findings I mentioned previously are consistent with the findings of Liu et al., who found the presence of both bat SARS coronavirus antibodies and human SARS coronavirus antibodies in Rhinolophus sinicus bats using a method called ELISA, or enzyme-linked immunosorbent assay. Detected bat SARS coronaviruses in 23 of the 59 samples from bats using RT-PCR, which is a direct detection method, as I said before, as opposed to the antibody tests, which are basically exposure tests to see if they have been exposed, rather than if they currently have the virus. So direct detection of the virus was done with RT-PCR, and some of the viruses, some of the PCR products, they ended up sequencing, and they ended up pulling phylogenetic analysis on, where they found that they reached the same conclusions as the previous papers I talked about, that SARS-related coronaviruses are human SARS-related coronaviruses are closely related with bat coronaviruses too. And they also added that bat SARS coronaviruses from a separate group together with the original human SARS coronavirus. And they classified that group as group 2b coronaviruses, which had a more distant phylogenetic relationship with the well-known group 2 coronaviruses. So group 2 coronaviruses was a classification that existed prior to the epidemic, and these researchers found that bat coronaviruses and the original human SARS coronavirus form a group that they named group 2b. And it was originally proposed to be renamed group 2. It was proposed to be renamed group 2a, so that it could accompany group 2b. And that is consistent with the previous findings I mentioned and some other findings from the years prior to 2005, like Gobrelenia et al. From 2004 and Snyder et al. 2003. The history of the origin of SARS coronavirus was solved in 2017 by Hu et al., a Chinese scientist group, after a 5-year study of SARS-related coronaviruses from bats living in a cave in the Chinese province of Yunnan. I'll underline this province because it's quite important and comes up a few times, so I want you to remember the Yunnan province. And they found a group of bat SARS-related coronaviruses with high genetic similarity to the original human SARS coronavirus. and terminal domain. This is for folks who know their molecular biology, they're nerds here. Some details for the nerds. If you don't understand this, it's okay. Just keep in mind that they found that bad SARS coronaviruses are similar to the original SARS coronavirus that caused the 2002 SARS outbreak. And they concluded that the progenitor of the human SARS coronavirus arose from recombination events that occurred among the identified bad SARS coronaviruses. And that was confirmed by the, well, not confirmed, but supported by their recombination analysis data. And that's also consistent with the discovery that several bad SARS coronavirus strains with different spike proteins circulating in bats can infect human cells via the ACE2 receptor, and they can replicate in them. With they confirmed that with RNA quantification by RT-PCR, which is basically a direct detection method. So they directly detected that were able to infect human cells in vitro. Basically, they put bat coronaviruses in a human cell culture, and they saw that bat coronaviruses could infect human cells, which is quite interesting. And that pretty much all this information added by the paper and previous publications pretty much leads to the conclusion that, oh, wait a minute. I'm back. I'm sorry if you had my doorbell ring, but I had to attend. Anyway, I'm back now. And what was I? Oh yeah, so this paper, along with the previous papers, pretty much led to the conclusion that the human SARS coronavirus had originated from bats, or at least the virus that gave rise to the human virus had originated in bats. So all the evidence put together pretty much conclusion, as I said, the building blocks for the human SARS coronavirus were found in bat SARS coronaviruses, and they concluded that the natural host of SARS coronavirus is the bat of the species Rhinolophus sinicus, since SARS-related coronaviruses with high homology to the human virus were detected, were mainly found in that species. So now we're going to talk about the well-known COVID-19 outbreak and the initial investigation when it started. Right, so on December 26, 2019, so in late 2019, a patient presented with symptoms of pneumonia to the Central Hospital of Wuhan, Hubei Province, China. The patient tested negative for pathogen causing common respiratory diseases such as influenza, chlamydia, mycobacterium, etc. So known pathogens weren't detected in that patient. By radiological findings confirmed they had pneumonia. Radiological findings meaning X-rays and CT scans. So by December 29, a few days after that initial case, 4 other patients had been hospitalized with similar clinical findings, with pneumonia and similar symptoms. And they were also negative for common pathogens that cause respiratory tract infections, which hints that there may be a new disease because all the pathogens we know weren't detected in those patients, all the relevant respiratory pathogens. And epidemiological investigation revealed that they all visited or worked at a seafood market selling several exotic animals in addition to seafood. So that market is called the Huanan Seafood Wholesale Market, and it's located in Wuhan City. In the same city, all 5 patients were from, and that prompted the hospital to alert the Wuhan Centers for Disease Control, which detected more patients who had visited that seafood market and linked all the patients to the market. Most of the early patients were linked to the market. And that was achieved using the pneumonia of unknown etiology surveillance system, which interestingly, it's the Chinese system that was established in response to the 2000 SARS outbreak in order to track emerging respiratory infections. So what the Chinese did after probably the Chinese just read the ISIS messages. But yeah, what the Chinese did after the 2002 SARS outbreak, they were smart and they said, hey, let's set up a surveillance system that tracks for pneumonias of unknown etiology, so that if another outbreak of a novel pathogen starts, we can detect it easily. That's what they did. That's why this new outbreak of COVID-19 was detected pretty much not immediately, but it was detected pretty early. And you'll see even the virus that causes it was found pretty early in comparison to the SARS outbreak. So December 30, that was all within the span of like 5 days. So on December 30, the local Wuhan Centers for Disease Control and Prevention notified the national Chinese CDC, because Chinese health system have city CDCs. For example, Wuhan CDC, they have provincial CDC for the province. For example, Hubei is the province that Wuhan is located in, and then they have a national CDC, a nationwide CDC, so citywide, province-wide, and then nationwide. So they alerted the nationwide CDC, which sent scientists for further investigation, where they took clinical samples from the patients for lab testing. After sequencing and analysis of the RNA they extracted from a lung sample of the patient, specifically to bronchoalveolar sample, I included that detail for the nerds. For anyone who doesn't know what a bronchoalveolar sample is, it's basically a lung sample. It's from an area of the lungs. And through metagenomic techniques, sequenced approximately 30,000 nucleotides long, had high homology to a previously sequenced bat coronavirus was identified. So that tells us a few things right off the bat. There is a virus that was detected from a patient that is very closely related to a bat coronavirus. That's quite interesting, and it gives us some clues as to where the disease originated, or that cluster of patients, how it came to be. So the genetic sequence of the novel virus was verified by RT-PCR, and the virus was named WH human 1 coronavirus. So W comes from Wuhan, so Wuhan human coronavirus. Phylogenetic analysis showed that this novel virus belongs to the genus Beta coronavirus and the subgenus Sarbecovirus of the Coronavirus family, and that it is highly related to SARS-related coronaviruses, especially bot SARS-related coronaviruses. So you'll see here I have included a phylogenetic tree for anyone interested in further details. If you can see my cursor here, here is the novel virus isolated from the original patient that had a typical pneumonia. And here are the two closest viruses related to it. They are bat coronaviruses. And here are some other coronaviruses that are related to it. You can see most of the closer related ones are bat coronaviruses. So that is quite interesting, and it might give us some clues about the origin of that virus. By mid-January, more cases had been identified based on a clinical definition by the Chinese public health authorities. By January 20th, the number of cases had reached 201. It's classified this event as an epidemic of a typical pneumonia. So they still weren't sure of the cause. The cause wasn't confirmed because of this new epidemic wasn't confirmed. But a common symptom that was shared by most patients was pneumonia, and the radiological findings like the x-rays and as well as the symptoms like cough and chest pain, all those were consistent with viral pneumonia. So all those findings they were hinting at it being viral in origin as compared to bacterial or parasitic. So the Chinese CDC confirmed that the newly discovered coronavirus that was named 2019 novel coronavirus by the World Health Organization, it was the causative agent of the recent pneumonia outbreak. So it was confirmed that the new virus is the one that's causing this pneumonia epidemic. So the Chinese CDC also reported the discovery of 15 environmental samples that were collected in the west wing of the Huanan seafood market that tested positive for this novel virus. So Zhu et al. In 2020 detected the new virus in multiple patients, most of which were linked to the Huanan seafood market. Not only verified the above laboratory findings that I mentioned, but also found the origin in the partially sequenced RNA-dependent RNA polymerase gene, which is a gene involved in viral replication. Just if you're not familiar with what that is, just remember the origin in a gene from a bat coronavirus sample that was named RATG13, which is important. It will come up a few times, that name, so I suggest you try to remember RATG13. That was what the sample was named. Also keep in mind it was previously named bat coronavirus 4991. That was the name of the sample. So when it was originally collected, it was named bat coronavirus 4991, sample 4991, and it was collected in 2013 from a Rhinolophus bat, or Rhinolophus affinis bat, in Yunnan province. You see this Yunnan province come up a lot, and it's important. So that sample had high homology with the novel coronavirus. This is a phylogenetic tree from the paper I mentioned before from Zhu et al. 2020, and you can see that this red thingy here is the new human coronavirus that was found to be responsible for the COVID-19 pandemic. And this here is the most closely related virus to the human one. And you can see it's a bat coronavirus, which is pretty interesting. And it gives us some clues to the origin of the new virus. And you can see all the other closely related viruses are also from bats. So that is pretty interesting. These are the human ones. If you can see SARS-CoV-2 BJ01, that is a human coronavirus from the original SARS outbreak of 2002. And all those more closely related ones are from bats. That's quite interesting. That's where the bat theory came from because, well, we see that it's very closely related to bats coronaviruses. So it was proposed that it came from bats. That's how the building blocks of the theory started. Sequencing the rat G13 sample, which is, keep in mind, the closest relative to the virus that causes COVID-19. And after comparing it with the virus that causes COVID-19, the two sequences were found to be 96.2% homologous. And phylogenetic analysis based on the S gene and the RDRP gene and the full genome so that rat G13 is the most closely related virus to the causative agent of COVID-19. And they form a distinct lineage that distinguishes them from other SARS-related coronavirus. And at the same time, the show officially named the disease COVID-19 because it didn't really have an official name up until then. And sometimes people referred to it as COVID-19 pneumonia because the early patients had pneumonia in common, had that symptom in common. And the novel virus that's responsible for COVID-19 was officially named SARS-CoV-2 because phylogenetic and genetic analysis showed that it belonged to the same species as the original SARS coronavirus. So it was taxonomically categorized to include that relationship. So in the following months, the existence of SARS-related coronaviruses was discovered through metagenomic analysis in pangolins, and those were given the name of pangolin coronaviruses, which showed high sequence similarity to SARS-CoV-2 and its closest relative, rat G13. But they also showed high similarity in the amino acid sequences, not just the nucleotide RNA sequences, but also the amino acid sequences that make up the proteins. And here are more details if you're interested. You see, the E protein was completely the same between all those viruses, but that doesn't tell us much because the E protein isn't really variable and it doesn't change much among coronaviruses. The one that changes the most is the S protein because that's involved in receptor binding, and so there's evolutionary pressure for that to continue to change to make the binding more efficient so that the virus can infect cells better and replicate more efficiently. So the S1 region of the pangolin coronavirus is more similar to that of SARS-CoV-2 than to rat G13. And for here, I mistakenly included like the reference. And the receptor binding domain of the S protein of pangolin coronavirus was exactly the same as that of SARS-CoV-2, including the 6 major amino acid residues that are involved in the binding. But there was only one amino acid different between the pangolin coronavirus RBD and the human coronavirus RBD. from the 193 amino acids, they had only one difference. So that is quite interesting, because RBD is like the most variable region of all the proteins. The RBD, which is a part of the spike protein, is quite variable, and it changes from species to species, and even across human variants of the same virus, it changes quite a lot. For the reasons I mentioned earlier, for evolutionary reasons that promote viral replication, viral entry to the cell. So nevertheless, rat G13 is still the most phylogenetically related virus to SARS-CoV-2, and the data so far suggested that it was probably of zoonotic origin, probably from bats, because it's related to many bat coronaviruses, and there's a possibility there is an intermediate host. Now we're going to go over some theories that have been proposed that attempt to explain the origin of SARS-CoV-2, which is the causative agent of COVID-19. So for the people who weren't here before, I said that I like to make the distinction between disease and the pathogen that causes the disease. So COVID-19 is the disease and SARS-CoV-2 is the pathogen, well, the virus that causes it. People confuse the two, but a distinction shall be made, and it's quite useful in an academic context. The implications being that pathogens need not induce the disease. Well, that too, but the implications are both that, but also that there are many pathogens that are related to each other, and saying their taxonomic name is important in order to distinguish between either variants of the same pathogen or closely related pathogens of other species, potentially. So that was in response to Ice's comment. Alright, so theories that regarding the origin of SARS-CoV-2. I'm just going to refer to it as SARS-2 for shortcut, because it's saying the entire thing over and over again. So there are alternative theories regarding the origin of SARS-2 that reject the zoonotic origin, and they have been proposed since the beginning of the pandemic, since it was, popularized by the media in early 2020, as many of you remember. Those theories were mainly proposed by people who lack scientific training, and yes, I do have a reference for that if anyone wants it. One of these is the theory that SARS-CoV-2 is the result of genetic engineering. Surprise, surprise. Everyone has heard of that. This theory doesn't seem to be compatible with the scientific evidence we have so far, and many papers supporting it, such as that of Yan et al. 2020, are based on unsubstantiated claims that are not consistent with the current data. For example, one of their arguments is their questioning. Oh wait, I missed it. It's their questioning of their validity of RAG13, and they're going as far as to question its existence, even though there's evidence that the sample was collected in 2013. It was then called bad coronavirus 4911, as I mentioned earlier, and that's important because you'll see later why that's important. A partially sequenced region of its genome was published in 2016, years before the COVID-19 pandemic, as mentioned above, and this sequence is 100% homologous only with the recently published fully sequenced RAG13 genome, with no other sequence. that tells us that there was a genome published in 2016, not a complete genome, but just a partially sequenced gene, and it only matches to rat G13. That was published, keep in mind, before the pandemic, before COVID-19, and it's referenced. It's referenced before the pandemic. So people saying it that rat Z13 is completely made up to support the zoonotic origin of of the virus, that is just nonsensical, given that it's mentioned even before the pandemic started in 2016. And if you see the gene that was published in 2016, it only completely matches to G13, no other virus. So we can conclude that it belongs to the rat G13 genome. And it wasn't made up because phylogenetic and genomic data are consistent with it being a sequence found in nature rather than something that was artificially created fraudulently. And that makes the criticisms regarding its existence as a sequence unfounded. If you go back to the paper published in 2016 and gene, a database of, all coronavirus genomes, it only matches to rat G13. And as I said before, and a lot of people doubt the existence of rat G13, but it was previously mentioned before the pandemic as bat coronavirus 4991. It just so happens that they changed the name of of that virus, of that sample, the sequence, to reflect the year it was collected in, which was 2013, and the bat species it was collected from, which is Rhinolophus affinis. That's the reason they changed the name, and a lot of people are confused and think that it was just made up as an excuse to support the zoonotic origin, but it was mentioned even before COVID-19 was a thing. So what if the animal sources were bred in the lab? That's what IC asks. I investigate that hypothesis later on, so stay tuned. Let's see. Right, so moving on to the next slide. Other claims, Yan et al.'s claims supporting their theory have been debunked by analyses of the SARS-CoV-2 genome, which have found no trace of genetic manipulation, further supports the zoonotic origin theory. Because Yan et al. Claim that SARS-CoV-2 is genetically manipulated, but Anderson et al. 2020 so that claim is completely unfounded, and there is no evidence of genetic manipulation in SARS-2 genome, in the SARS-2 genome. There is no evidence of genetic manipulation either by CRISPR or other restriction enzyme methods that are used in genetic manipulation. And an essay published by the Bloomberg School of Public Health of Johns Hopkins University deconstructs the Yale et al. Preprint in more detail, so I'm not going to bother talking about it further. So some other scientists, such as Segrito and Dagin, have suggested that the existence of a furin cleavage site, which is abbreviated as FCS, in the SARS-CoV-2 genome suggests that the virus has been genetically engineered. This claim fails to acknowledge the following facts. One, the fact that the role of the FCS in the coronavirus genome was not well understood before the COVID-19 pandemic. there would be no reason for someone to deliberately insert it into the SARS-2 genome. To the fact that the FCS is much more likely to occur under evolutionary pressure and immune selection, which Segreto and Dagian ironically admit in their paper, and that the FCSs have evolved multiple times independently in multiple coronavirus lineages during their evolution. This was proven by Wu and Zhao in a 2020 publication, which makes the presence of a furin cleavage site in SARS-CoV-2 consistent with the zoonotic origin and not more consistent with the genetic manipulation theory. Segreto and Dagian acknowledge and describe an event that might be an important clue regarding the origin of SARS-CoV-2. In 2012, 6 miners were hospitalized with atypical pneumonia. I mentioned it before, but for the people who weren't here, atypical pneumonia means pneumonia that is caused by a pathogen that we don't know yet, like by no known pathogen. At a hospital in Yunnan province, which is a really interesting province of China that will come up a few more times during this presentation. 3 of the patients died. A few days before they were hospitalized, these miners had been tasked with cleaning bat feces at a mine in Mojian County, which is located in Yunnan province as well. This incident was originally described in a master's thesis by a student called Zhu in 2013, which concluded that the etiology of the atypical pneumonia cluster in the miners was a bat SARS coronavirus, and that conclusion was derived from the clinical and epidemiological data presented in the thesis. Later, a team of researchers from the Wuhan Institute of Virology were sent to investigate the mine and look for agents that could have caused this cluster of atypical pneumonia. And they discovered that the mine was home to a large number of bats, particularly Rhinolophus bats, otherwise known as horseshoe bats, from which they took most of the samples. They discovered 150 different strains of the genus Alpha coronavirus and 2 strains of the genus Beta coronavirus, one of which was a SARS-related coronavirus and was named Rat coronavirus 4911, also known as RATG13. So you see a familiar sequence come up. RATG13, that is its origin story. It was collected from a mine in Yunnan province. From a bat sample from a mine in Yunnan province. And it was a sequence generated through metagenomic processing. That is quite an interesting origin story for the closest yet known relative to SARS-CoV-2. Originated in Yunnan province. So if you were here earlier, you remember that Yunnan province is the province where SARS, the progenitor to the original SARS coronavirus that started the 2002 SARS outbreak, was found in Yunnan province. Bats of genus Rhinolophus. So that's quite interesting. So in 2018, before the COVID-19 pandemic, Wang et al. Published a paper describing their research using serum samples they took from 218 residents of villages in Yunnan province. Again, this very interesting province in China that comes up quite often, Yunnan province, that was located near caves and inhabited by Rhinolophus bats. The horseshoe bats are favorites, in which SARS-related coronaviruses have been previously found. And they compared those samples to a control group of 240 blood donor samples from Wuhan city. Ironically, the city were 1 year later after the study was published, the COVID-19 pandemic started. And they found that 6 SARS positive samples from the villagers in Yunnan were positive for SARS using ELISA, which is an antibody detection method, a serological method. But no one from the control group of blood donors in Wuhan were positive. And this is, if you're interested in more detail, here's the figure that was included in the paper. It shows the cutoff point, which is the dotted lines, and it shows the samples that tested positive. So serum samples taken in 2015. The study was conducted. It was published in 2018, but the serum samples from the Yunnan villagers were taken in 2015, which is more than a decade after the 2002 SARS outbreak. And that is important because antibodies from the 2002 SARS virus last about a year or two. So it is unlikely that people in 2015 would have antibodies from exposure from the 2002 SARS virus. So considering the low seroprevalence of SARS antibodies in the region, due to the small number of SARS cases in the original 2002 outbreak, it was about 8,000 cases, so it wasn't a huge number of cases. It wasn't spread across China, it was in a few distinct provinces, not in Yunnan, in other provinces like Guangdong. So the significant decline in antibodies, as I mentioned earlier, after about 2 years after exposure to the original SARS pathogen, and the lack of contact of the study participants with SARS patients, all suggests that the 6 positive samples for SARS antibodies in Yunnan residents were from infections after the 2002 SARS epidemic, which means there might have been a more recent undocumented spillover event from bat SARS coronaviruses that are responsible for the antibodies seen in the study. Because antibodies, the fact that they had SARS antibodies means that they were exposed to a SARS-like virus. So that is quite interesting because that was after the original SARS epidemic and before the COVID-19 epidemic. So Frutos et al. Published a paper in which they present arguments against the laboratory origin of SARS-2, and they deconstruct a lot of the claims supporting that theory, the laboratory theory. Also noting that the clinical findings and symptoms reported by Zhu in his master's thesis that I previously mentioned regarding the 6 cases of minors pneumonia are not consistent with those of COVID-19. So Frutos here raises the argument that Zhu's conclusion about the minors in Yunnan are wrong and that their symptoms are not consistent with COVID-19. And that's also supported by the fact that their serological samples tested negative for SARS-2 antibodies when tested by Zhu et al. In 2020 in the initial paper describing the then recent COVID-19 pandemic. They also explain, Frutos et al. Also explained that it is not likely that the COVID-19 pandemic originated from workers from the Wuhan Institute of Virology who got infected. at the Mojiang mine during their investigation, because the causative agent for the COVID-19 disease, which is known as SARS-2, has not been identified at the mine, and WIV employees have been found negative for COVID-19 when tested as part of an investigation. So it is unlikely that SARS-2 was started by Wuhan Institute of Virology workers. It is tested as part of an investigation that were negative initially, in the early stages of the pandemic. There wasn't a high prevalence of SARS-2, and SARS-2 hasn't been discovered in the cave where the miners worked. Maybe the virus didn't originate in that cave. Also highlight that the previously mentioned rat G13 is not a virus that has been isolated or cultured, so it's not a viable virus that we have cultured. It's a sequence that was obtained through metagenomic analysis of a bat sample from that cave. There's a possibility that this sequence does not correspond to a single viable virus. It may have arisen from multiple viruses due to limitations in metagenomic analysis techniques. Even if the rat G13 sequence does correspond to a viable virus that has been isolated and cultured, the significant difference between the genome of rat G13, which is the closest relative to SARS-2, and all other known coronaviruses, including the ones that were used for gain-of-function research in the Wuhan Institute of Virology, and SARS-2, as well as their phylogenetic distance, both show that RAD-Z13 is not the progenitor of SARS-2. So RAD-Z13 and SARS-2 are too far apart for SARS-2 to have originated from RAD-Z13. You can't say SARS-2 originated from RAD-Z13 as they're too phylogenetically apart. Even though they're the closest, RAD-Z13 is the closest relative to SARS-2. Therefore, it's impossible that SARS-CoV-2 has been derived from RAD-Z13 in a laboratory environment, for example, in gain-of-function research as supported by some people, or through genetic manipulation of RAD-Z13, or any other known coronavirus, because SARS-2 is just too far apart from other known coronaviruses to be derived from them, thus debunking the claims that SARS-2 is the product of gain-of-function experiments at the Wuhan Institute of Virology. So in a subsequent paper published 2 months later by Frutos et al. Again, they proposed a model that describes the zoonotic origin theory of SARS-2 without the need for an intermediate host. It has some differences compared to the classical proposed zoonotic spillover model. They call the circulation model, based on ecological, genetic, molecular, and evolutionary data. This is a schematic representation of the model they proposed. I'm not gonna explain it fully because this is not the purpose of the presentation, but they basically propose a model virus. This virus was circulating in animals, and the animal-human interface, and at some point there was evolution within human hosts that led to a virus, an evolved version of the virus that gave rise to the COVID-19 pandemic. Rather than a direct spillover event from animals to humans, there was circulations from animals to humans, and that resulted to a virus evolving in humans and giving rise to the pandemic. That's what they propose, but it's obviously not confirmed yet. So there was a recent paper published in Nature, which is a really prestigious scientific journal for those who haven't heard of it. It was it was authored by scientists at the Pasteur Institute in collaboration with scientists. And the paper was led by Dr. Tamam. So the samples were collected from bats from a cave in Laos, which is a country that borders Yunnan province in China, and they discovered an isolated bat SARS-related coronaviruses with an RBD similar to that of SARS-CoV-2. Allows entry into human cells via the same receptor that SARS-2 uses, which is ACE2. They fully sequenced several bat SARS coronaviruses, including a virus they named BANO52, from a Rhinolophus bat. This is now the closest known relative SARS-2, and it has a 96.8% similarity at the genome level, and it replaced rat G13 as the closest relative to SARS-2. It only had 6.2% sequence similarity to SARS-2. Phylogenetic analysis they performed placed BANO52 on a couple of other viruses that they isolated from Rhinolophus bats. It placed them close to SARS-2, rat G13. All of these are closely related viruses. Keep in mind that they're all closely related. And I've included a picture to show geography of the area they were working at, because that's kind of important here. So you see this tiny country called Laos. It borders China here, if you can see my cursor. And this part of China is actually Yunnan province. Interesting. And these are the sites they collected the bat samples from. They are caves in Laos with pursue bats. And this is Yunnan province, so this country, Laos, and some of the sites they sampled the viruses from. Here, it's very close to Yunnan province. There's some clues about the origin of SARS-2 or its progenitor. So this is a phylogenetic tree based on whole genome analysis of the genome of SARS-2 and other related coronaviruses. You can see that here it's the original reference genome of SARS-2. Here is rat G13, here is BANO52, here are some other bat coronaviruses that they sampled in that research in 2022. Well, it was published in 2022, but it was done in 2021, late 2021, and you can see some other viruses, like you can see pangolin viruses. They aren't as closely related to SARS-2 at the whole genome level, but you can see how the human SARS-2 pathogen is closely related to bat coronaviruses, which is why a lot of scientists propose that it has a zoonotic origin specifically from bats. That's what the bat theory is based on. So, as the authors point out, bat SARS coronaviruses that resemble each other are present in many bat species of the Rhinolophus genus, and that suggests that these viruses circulate among different bat species of the Rhinolophus genus that share the same caves, and that is consistent with the fact that BANO52 and rat G13 show high genomic similarity to SARS-2. However, SARS-2 shows higher similarity to BANO52 than to rat G13, the S1 gene, especially the RBD, which is the most variable part of the whole genome, the whole bat coronavirus genome. And that is also reflected in the amino acid sequence. So not only the gene, but also the translated protein derived from the gene also have similarity from between Banal 52 and SARS-2. So in both the genome and the protein level, they are similar in that regard. Specifically, 17 amino acids from the receptor binding domain that interact with the human ACE2 receptor. SARS-2 shares 16 amino acids with Banal 52, so 16 of the 17 key amino acids that interact with the human ACE2 receptor. And wait, and Banal 103 also shares 16 amino acids with SARS-2, 16 of the 17. While Banal 236 also shares 15 of the 17 amino acids with SARS-2, and RATG13 only shares 11 amino acids of the 17 with SARS-2. So in silico, which means in computer simulations, molecular dynamic simulations examining the receptor binding domain and the human receptor complexes of the bat coronavirus RBDs, they showed interactions between the RBDs of Banal and the human receptor, and they are similar to SARS-CoV-2. So the molecular dynamics of the bat RBDs and the human receptor are similar to the molecular dynamics of the human RBD, of the human virus RBD, and the human receptor, which means that the mechanisms by which the bat viruses bind to the human receptors are similar to those of SARS-2. This is a figure that shows the similarity of the different coronaviruses, and it's categorized by the gene, basically, or well, the protein that the gene codes for, and it is on the amino acid level, not on the genome level, so on the translated protein level. You can see that SARS-2 and Banal 52 are very similar, and RATG13 is not as similar as Banal 52 is with SARS-2. This figure was taken by the Tamam et al. Paper, and the data that have been presented in the paper are consistent with the findings of in vitro experiments that this research group did, where they generated lentiviral particles that expressed the proteins, the S proteins of Banal 236, so bat coronavirus, and they detected the entry of those viral particles in human kidney cells that expressed the ACE2 receptor. They didn't detect entry of the virus into control cells that didn't express the human ACE2 receptor. That means that the S protein allowed the bat coronavirus S protein allows entry into human cells in vitro, in a laboratory environment. So they also found that neutralizing antibodies against SARS-2, entry of the viral particles that expressed the bat coronavirus proteins into human cells. So that is quite interesting. Make of that what you will, but those are the current data we have until the present day. Now, concluding remarks. Coronavirus epidemics aren't new. The first coronavirus epidemic was in 2002 with the original SARS outbreak. The virus that caused that epidemic was shown to be of zoonotic origin, specifically from bats in Yunnan province, from horseshoe bats in Yunnan province. SARS-like coronaviruses have been known to circulate in bats, since bats are the natural hosts of SARS-like coronaviruses for more than a decade and a half. So the paper that first suggested that was published in 2005. Most emerging infectious diseases are of zoonotic origin. The origin of SARS-CoV-2 is still unknown, so we don't have a definitive conclusion, but there are data that suggest that the origin is zoonotic. The SARS-CoV-2 genome and phylogeny is consistent with the zoonotic origin, and it's inconsistent with the man-made or bioweapon theories, as the virus appears to have evolved naturally and not have been artificially created in a lab, especially since there is no evidence of genetic manipulation. So the closest relatives to SARS-CoV-2 are coronaviruses from bat samples from a region that's geographically close to the region of the original SARS-CoV-1, where the progenitor of the original SARS-CoV is believed to have originated from, which is in general the Indo-Chinese Peninsula, the region where Yunnan province of China and Laos belong to. So finding the origin of SARS-CoV-2 will hopefully lead to better public health interventions and disease surveillance and prevention and control. It will help address future outbreaks. These are some of the references I used for the presentation, though too many references to post here in the presentation. There are like 67 referenced papers, and there's about 100 papers in total that I read in order to come up with this data. So these are some of the most important papers that are referenced. If you want a full list of all the 67 references that I used in my original paper that I based the presentation on, you can DM me and I will provide it to you. So these are some of the references that I used. You can see the Frutos paper, the Tamam paper, they're all here. And that's about what I have for today. I hope you enjoyed the presentation. If you have any other questions, you can post them in the voice channel. I'll try to answer them in like a short Q&A section that we'll do now that the presentation is over. And if you have any further inquiries, you can DM me about it.